Hi everyone, welcome to all. This is Vidyam Academy's NEET Initiative Most Expected Question Series. This is Rajiv Patel. Today's topic is 12th Biology, 4th chapter, the principles of inheritance and variation. Into this, subtopic is inheritance of two genes and sex determination. The first question is, Mendel crossed pure round yellow RRYY piece with a pure wrinkled green RRYY piece. What phenotypic ratio did he observe in the F2 generation of this dihybrid cross? So here the options are option A, 9:3:3:1, round yellow, round green, wrinkled yellow, wrinkled green. Option B, 1:1:1:1, equal proportion of all four phenotypes. Option C, 3:1, same as monohybrid cross for each trait independently. And option D, 12:3:1, modified ratio due to ephistasis. What is the correct answer for this question? Option A. A is correct answer for this question. 9:3:3:1, round yellow, round green, wrinkled yellow and wrinkled green. Why and how? Let's see the explanation. In a dihybrid cross, P generation RRYY into RRYY gives all F1 a round yellow, both dominant. When F1 RRYY plants self-fertilize, each gene independently assorts into gametes. So here RRYY segregates separately, giving 4 gamete types in equal proportions. The 4x4 Punnett square produces 16 combination with phenotypic classes. So 9 round yellow, 3 round green, 3 wrinkled yellow and 1 wrinkled green. This ratio 9:3:3:1 is the hallmark of dihybrid cross with independent assortment and proves that the two gene pairs segregates without influencing each other. Each trait individually still shows 3:1, confirming both traits follow the law of segregation independently. Let's move to question number 2. Mendel's law of independent assortment applies to which situation and what does it state? Option A. Applies when two genes are on the same chromosome, they always assert together. Applies two genes on different non-homologous chromosomes, alleles of different genes assert independently into gametes. Applies to all genes regardless of chromosomal location, even linked genes assert independently. Option D. Applies only to homozygous organisms. Heterozygotes do not show independent assortment. What is the right answer? Option B is right answer. Let's see how and why. The law of independent assortment states that during gamete formation, the alleles of one gene segregate independently of the alleles of another gene, provided the two genes are located on different non-homologous chromosomes. This happens because during meiosis 1, homologous pairs line up on the metaphase plate independently of each other. The orientation of one bevelent does not influence another. Mendel's 7 traits happen to be on different chromosomes, so all showed independent assortment. So when genes are on the same chromosome link, this law doesn't hold. They tend to be inherited together unless crossing over separates them. Independent assortment in the reason genetically diverse offspring are produced. That's what option B is the right answer. Applies two genes on different homologous, non-homologous chromosomes, alleles of different genes assert independently into gametes. Question number 3. In humans, sex is determined by the sex chromosomes. Which of the following correctly describes the human sex determination mechanism? Option A. Females are XY and males are XX. The Y chromosome carries female determining genes. Option B. Females are XX and males are XY. The Y chromosome carries that SRY gene that determines maleness. Option C. Sex is determined only by the number of autosomes, not by sex chromosomes. Option D. The females are XX and males are XO. Males have only one sex chromosome. What is the right answer for this question? The right answer is option B. Let's see. Females are XX and males are XY. The Y chromosome carries the SRY gene that determines the maleness. In humans, females carry two X chromosomes. We can call it as XX. And males carry one X and one Y chromosome, XY. This is the XXXY system. The Y chromosome carries the SRY gene, sex determining region Y, located on the short arm of Y, which acts as the master switch that directs the undifferentiated gonads to develop into testes. Without SRY, ovaries develop by default. So females produce only X-bearing eggs, homogametic sex, while males produce 50% of X-bearing and 50% of Y-bearing sperm. So heterogametic sex. When a Y-bearing sperm fertilizes the egg, a male XY results. When an X-bearing sperm fertilizes the egg, a female XX results. Sex is therefore determined at fertilization and depends entirely on which sperm reaches the egg. That's what. So here the determination of the sex depending upon by the females are XX and males are XY. The Y chromosome carries the SRY gene which determines the maleness. Let's move to question number 4. In grasshoppers, which sex determination mechanism operates and what chromosome constitution do males and females have? Option A. XXXY system, females are XX, males are XY, same as humans. Option B. ZWZZ system, females are ZW, males are ZZ. Option C. XXXO system, females are XX, 44 plus XX, and males are XO, 44 plus X, having one fewer sex chromosome. Option D. Haploid-diploidy system, females are diploid, males are haploid. What is the right answer for this question? The right answer is option C. Let's see why and how. Grasshoppers use the XXXO sex determination system, one of the simplest chromosomal mechanisms. So females are XX, they carry two sex chromosomes in addition to their autosomes. So males are XO, they carry only one X chromosome and have no Y chromosome at all. The word denotes the absence of second sex chromosome. So during meiosis, 50% of male gametes receive the X chromosome and 50% receive no sex chromosome. When an X-bearing sperm fertilizes an X-bearing egg, the result is XX female. When an O-bearing sperm fertilizes the egg, the result is XO male. This differs from the human XXXY system, so where males have a Y chromosome. So in grasshoppers, there is no Y and maleness results simply from having only one X. And next move to question number 5. In birds, which sex is heterogametic, produces two types of gametes, and what is the chromosomal constitution of each sex? Option A. Males are heterogametic ZW, females are homogametic ZZ. Option B. Females are heterogametic ZW, males are homogametic ZZ. Option C. Both sexes are homogametic ZZ for males and ZZ for females. Option D. Birds follow the same XXXY system as humans. Females are XX and males are XY. Would you guess the right answer? Your time starts now. Option B is right answer. Why and how? Let's see. So birds use the ZWZZ sex determination system, which is the reverse of the human XXXY system. In birds, males are ZZ homogametic. They produce only Z-bearing sperm. And females are ZW heterogametic. They produce 50% of Z-bearing eggs and 50% of W-bearing eggs. When a Z-bearing egg is fertilized by Z-bearing sperm, a ZZ male results. When a double-bearing egg is fertilized by Z sperm, a ZW female results. So in birds, unlike in humans, it is the mother's egg, not the father's sperm, that determines the sex of the offspring. This ZWZZ system is also found in some reptiles, fish, and butterflies. The key distinction from the human system, female is the heterogametic sex in birds. That's what. So this is the right explanation and the correct answer, option B. Question number 6. An organism with genotype AABB produces how many types of gametes and what are they? Assume both genes follow independent assortment. Option A, 2 types AB and AB only. Option B, 4 types AB, 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 AB in equal proportions. Option C, 2 types AB and AB in 3:1 ratio. Option D, 8 types because the organism is heterozygous for 2 genes. The right answer and option is option B, 4 gamete types AB, AB, AB and AB. So an organism heterozygous for two independently asserting genes. So form four types of gametes in equal proportions. So during meiosis, the A pair segregates A into one cell, A into another, and independently of this. The BB pair also segregates B of B. The four possible combinations are AB, 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 and AB. So each present at 25% frequency. So this is the direct consequences of the law of independent assertment. The two gene pairs segregates without influencing each other. The formula for the number gamete types is 2, where n is equal to number of heterozygous gene pairs. So n is equal to 2. So that's what. So here 4 gamete types. A homozygous organism produces only one gamete type. Let's move to question number 7. T.H. Morgan performed dihybrid crosses in Drosophila and found the certain gene pairs didn't show the expected 9:3:3:1 ratio. What was his explanation for this deviation? Option A. The genes underwent mutation during the cross, so changing the expected ratios. Option B. The two genes were located on the same chromosome linked and tended to be inherited together rather than independently. Option C. So Drosophila follows incomplete dominance for all traits, modifying the dihybrid ratio. Option D. The 9:3:3:1 ratio doesn't apply to animals. It's only valid for plants. What is the right answer? Option B is right answer. Why and how? Let's see the explanation. So Morgan found the certain phase of traits in Drosophila appear together in offspring for more often than expected from independent assertment. So deviating significantly from the 9:3:3:1 ratio. His explanation was linkage. The two genes were physically located on the same chromosome and were therefore physically connected. So tending to travel together into the same gamete during meiosis. When genes are on different chromosomes, they assert independently. So based on the Mendel's law. But when on the same chromosome, they are linked and co-inherited. However, linkage is rarely complete. So crossing over during meiosis 1 can break the physical connection between linked genes and create recombinant gametes, which appear at a frequency proportional to the distance between the genes of the chromosome. That's what. Two genes were located on the same chromosomes linked and tended to be inherited together rather than independently is the right answer. Next, move to question number 8. Consider the following statements about sex determination. Statement 1. In the XY type humans, males are heterogametic and females are homogametic. Statement 2. So in the ZW type birds, females are heterogametic JW and males are homogametic ZZ. Statement 3. In honeybees, sex is determined by ploidy. So fertilized eggs develop into diploid females, queens and workers, while unfertilized eggs develop into haploid males, means drones. Option A. Only statement 1 is correct. Option B. Only statement 1 and 2 are correct. Option C. All 3 statements are correct. Option D. Only statements 2 and 3 are correct. What is the right answer? Option C is right answer. All 3 statements are correct here. Statement 1 is correct because in the human XXXY system, males XY produces 2 types of sperm, X bearing and Y bearing. So males are the heterogametic cells, while females only XX produce only X bearing eggs. So females are homogametic. Statement 2 also correct because in birds ZWZZ system, the female ZW produces 2 types of eggs, Z bearing and W bearing. So the females is heterogametic. And the male ZZ produces only Z bearing sperm, so males are homogametic. The reverse pattern from humans. Statement 3 is also correct because honeybees use haploidy-ploidy. The queen can control whether eggs are fertilized. Fertilized eggs diploid. So 2N develop into females. Queens are workers depending on nutrition. While unfertilized eggs haploid then develop into males drones by pathogenesis. So all 3 represent distinct and valid sex determination mechanisms. That's what all 3 statements are correct, which are mentioned here. Question number 9. Assertion A. Linked genes do not follow Mendel's law of independent assertment and are therefore inherited together more often than would be expected by chance. So here the assertion A. So here the reason R. During meiosis 1, crossing over between non-sister chromatids of homologous chromosomes can separate linked genes and produce recombinant gametes, which is why linkage is usually incomplete rather than absolute. So option A. So both A and R are true and R is the correct explanation of A. Option B, both A and R are true but R is not the correct explanation of A. So A is true but R is false. And option D, A is false. Linked genes always follow independent assertion. What is the right answer? Option B is right answer because both A and R are true independently, but R is not the correct explanation of A. Let's see why and how. Assertion A is true because genes on the same chromosome are physically linked and therefore tend to be inherited together, violating independent assertment. So they show co-inheritance frequencies above what random chance would predict. So reason R is also true, crossing over between non-sister chromatids during prophase 1 of meiosis does break the physical connection between linked genes and thus produce recombinant, so non-parental gametes, which is why absolute linkage is rare. However, R doesn't explain A. Assertion A states that linked genes deviate from independent assertment because they are on the same chromosome, so physical co-location in the case. R describes crossing over, which actually reduces linkage by creating a recombinant, so making it a counterpoint to A rather than the explanation of A. Since both A and R are true individually, but R explains the exception of A rather than the cause of A, option B is right because both A and R are true, but R is not the correct explanation of A. The final question of this topic, question number 10, arrange the following steps of Mendel's dihybrid cross experiment in correct chronological order. P, first one, so pure RRYY round yellow and pure RRY wrinkled green P generation causes P. Q, all F1 plants observed as round yellow confirming dominance of both R over R and Y over Y. Option R, F1 RRYY plants self-fertilize. Yes. 4 types of gametes formed by F1, RY, 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 RY in equal proportion. T, 16 square Punnett grid constructed, 9:3:3:1. So phenotypic ratio obtained. U, so ratio confirmed as evidence for the law of independent assertment. So here the options are option A, PQRSTU, option B, PSQRSTU, option C, PRQSTU, option D, SPQRTU. What is the right answer? Right answer of this question is option A, which are correctly matched chronological order way. So PQRSTU. So everything is correctly matched in a Mendel's dihybrid cross experiment. Let's see. The cross begins with P, Q breeding parents plants RRYY, RRY crossed first. So Q follows immediately because all F1 offspring are RRYY phenotypically round yellow, confirming that R dominates R and Y dominates Y. R is next. The F1 RYY plants are allowed to self-fertilize, which is the step that generates the critical F2 data. So X explains what happens at the cellular level during F1 gamete formation. Each RRYY plant independently asserts its 2 gene pairs, producing 4 gametes types. So each at 25%. T follows, combining the 4 gamete types in 4x4 Punnett square generates 16 boxes representing all F2 combinations, which collapse into the 9:3:3:1. So phenotypic ratio. So U is the final interpretive step. This 9:3:3:1 ratio is accepted as experimental proof of the law of independent assertment. So here we explained 10 most important and expected questions for your upcoming NEET examinations. And next, I will come up with another topic which are very important for your NEET examination. Till so far, I would like to conclude this session with very well goodness. Thank you and thank you so much to all.